Welcome everyone. This is a late onset Tay Sachs research session, and my name is Mohammad Eichler, and I understand most of you have already met each other here this morning, and I see many faces I've known over the years, and some faces that are new to me, so I hope to get to know you in the coming days. The purpose of this session here is to introduce you to the clinical assessments that we will be performing in the next 2 days, and tell you why we're doing these sessions and how we got to this place and what motivates us to ask for your help in performing clinical research. So most of you have lived with late onset Tay Sachs or Sandhoff disease, either as an affected patient or as a caregiver, and know about this disease as an affected person for much longer than some of us who come to this as medical researchers. But I think right now we're at a very exciting time in terms of medical research, where there's a heightened awareness of the importance of a patient perspective upon the disease. And there's also new technology and new approaches and new drugs that are coming down the pipeline, that if we all come together, if we all band together in aligning the efforts, I think there's a lot of momentum to move forward. So just to highlight that, we're really a team of people who are all here working together, and some of the members of the team are here from different academic institutions, but also from industry. So maybe I thought, since I have a few people here, I'll just have them introduce themselves. I'll briefly say a word about myself. I'm a neurologist at Massachusetts General Hospital. I run a translational laboratory that's devoted to bringing new cures to single gene disorders like Tay Sachs and Sandhoff disease. And we're involved in gene therapy trials and other supplementation trials.
Okay, for those of you we didn't meet this morning, I'm Cindy Tift, and I'm trained as a pediatric geneticist. I have a small lab at the National Institutes of Health that studies Tay Sachs and Sandhoff disease in mouse models and in cell culture systems, looking at different kinds of potential therapies. I also conduct a natural history trial at the NIH Clinical Center for late onset patients, for juvenile GM1 patients, pretty much patients over the age of 3 or 15 kilos, because that's at the moment, the NIH Clinical Center won't let me bring in smaller children than that, but we're working on it. And my interest in Tay Sachs and Sandhoff goes back to 1995, when we made the Sandhoff mouse model, I think.
Good afternoon, everyone. My name is Alaa Hamid. I'm with Sanofi Genzyme. I'm part of the Health Economics and Outcomes group. I work closely with our R&D colleagues, especially Jerry Cox, who's going to be joining us later today. Part of our work streams that we're working on in Tay Sachs is trying to develop a patient-specific, patient-reported outcome measure, a disease-specific. Last year, we conducted a few focus groups to get your feedback on the impact of the disease on the patient, and also from a caregiver perspective. This work is going to continue, and the aim is to develop a scale or a measure that is patient-centric, that is specific for Tay Sachs.
Hi everyone, my name is Lily Haxton. I'm a research coordinator at MGH, and I work with Florian on starting up a natural history study in late onset Tay Sachs disease. So I'm looking forward to getting to meet all of you guys later this evening.
Hi, so I'm Jean Johnston. I've met almost everybody. I'm the research nurse for Dr. Cindy Tift at NIH.
Excellent. So I think you're seeing that there a lot of different people from academia and industry here. There are actually many more people involved in late onset Tay Sachs research that are not in this room. And I was actually just pulling up my email to actually make sure I was able to mention all the people, because our group has grown so much just in the past few months. And so just to mention, you mentioned Jerry Cox, who's from Genzyme, being really a wonderful contributor in helping pushing, I think, this next phase of research forward. Then I should also mention our most recent addition to our group, Christopher Stephen from Mass General, who's from the ataxia group, who's really studying the coordination problems due to cerebellar disorders, and bringing all this knowledge now to late onset Tay Sachs and Sandhoff disease. Then Swati Sathe, who's from Rutgers, who for many years has also been studying late onset Tay Sachs and worked with Ed Kolodny back in the day. And who else am I missing? Julie Kissel from Genzyme, of course, who's really helped to coordinate everything and make sure we're all on the same conference calls. And I will say it's been a fantastic effort on her part, really trying to get everybody's schedules to work out so that we're really coordinating all these calls and making sure that our outcome measures are aligned. And Heather Lau, who of NYU, who's also here and joined our first meeting 2 years ago at Mass General, and will be also part of the neurological assessments. Anybody else have forgotten? I think.
Well, just the people, my people that aren't people. One of the neurologists that works with me that's not here that is Camilla Toro, and I think part of the plan for this series of evaluations is we're also, thanks to Julie and several others, bringing video cameras, and with your permission, we'd like to videotape the neurologic exams so that we can actually have 5 different neurologists all do the same observation and the same kind of assessment, so we make sure that we're doing everything sort of correctly. And my speech therapist, I think, is also going to weigh in at some point, but couldn't be here today.
Good. So I think Cindy has already alluded to the first important point that we're making progress in, which is aligning and harmonizing all the testing that we're doing in different institutions. And why is that important? I think as we are preparing ultimately for treatment development and for a trial, which is like the common goal here, right? We all know that research is fun for those of us who are interested in biology, but ultimately what we want is to bring some tangible benefit forward for patients. You've probably been to conferences like this before, and you've heard researchers speak about the disease. And I think one of the things that we really want to see in our lifetime happen is treatments that work. And you may ask yourself, so what does that have to do with yourself? What can you contribute? And why is that not just the business of the researcher or the scientist? I think what we've found over the years of treatment development is that it's not only important to understand the biology, but also to understand what is meaningful to patients. You can change an enzyme or a chemical as much as you want. If it's not bringing about a clinically meaningful change to patients, then the FDA will turn away and say this drug cannot be approved. This drug is of no clear benefit to patients. And so we need to understand from you how this disease affects you, and at what stage in the disease you see a possibility for things getting better, and how can we in the outcome measures that we are using, that we are assessing, make that happen. So I was thinking of this session as being a place where we could start having a little conversation about that and telling you how how this has led us to these kinds of assessments that we're doing together with not only NIH and NYU and other academic institutions, but also Genzyme and hopefully others that are interested in drug development over time. So what does FDA actually look for when they're trying to have a drug be approved? They are looking for a drug that has to be safe. They have to know that that drug is not doing any harm to patients, that it is, that we understand what kind of dose is well tolerated, that the side effects that might occur outweigh, are outweighed by any possible benefit. And then how do we actually measure whether something works? Now you might say, okay, a drug that actually helps me, I don't need to know exactly how it works, I just need to know that it works. But FDA will go to anybody who's running a clinical trial or involved in drug development and say, how have you measured that this drug is of benefit? So you need to know how you can measure that benefit. Is the patient able to walk faster? Are they able to get up from a chair faster? Is there something about their coordination that now is better and more smooth? Is it that certain events occur less frequently? If you're more stable, maybe you're falling less often, right? Anything you can measure is important when you're going and seeking drug approval. And then from what we need the patient and the community involved is to tell us, is whatever we're measuring here clinically meaningful? Right? And something's getting darker and I'm feeling more in the spotlight. But time's up. So I think, in short, what we're trying to do these days is instead of approaching research in biology as we've done in the past, which is define the disease by the pathology, study the chemistry, study the genetics, is turn the whole thing upside down and say, let's start with the end in mind. Let's start with how do we get a trial going for this rare disease? And if we start with that end in mind, that we actually want to get a trial going, then we understand what's missing and where are the gaps that we need to fill. And these are the gaps that we're really trying to fill in these next 2 days when we're sitting down with you and performing assessments where your participation really makes a world of a difference. So I have here notes from Sue Khan because she told me I have to give this introduction, and there are, I think, some other important points that I should make. So while we do have certain approaches that we think are going to work down the line, gene therapy or gene correction, since we know what gene is here involved, or maybe substrate reduction, and we have companies like Genzyme who are involved in this field. Our approach is really not about one specific company or one specific drug. We think that if we can create outcome measures, we understand together through these assessments what is meaningful and measurable, that we can use that information and create a platform that can be shared with whatever drug maker or whatever trial comes along, okay? So the idea here is we shouldn't be reinventing the wheel every time we're trying to come up with a new drug and think about the trials anew. So part of what we've done over the years is I think with the first step, Cindy and we developed surveys a few years ago, and we said, well, we really have to understand how the disease progresses over time. And really the surveys it was fascinating to see the return on that and the kind of information that patients had mothers, family members stored, and the kind of detail that was available that really helped us understand how this disease progressed. This was not something you could find in the medical literature. This was really stored away in binders of mothers or in the kind of information that patients knew so well, but was not in the medical literature. So I think that was an important first step for us and made us realize that there are certain functions, such as stability of gait, certain functions such as coordination or speech, and how clear your speech is, that are of particular importance to patients. And if we can get more definition around those different domains, then we can maybe find medications and drugs that would work and we could measure change over time. And I think the big problem has been that we've not had good quantifiable things that we can actually measure to tell us whether a drug is working or not. Okay? But I think this is the one big gap we're trying to address here in these sessions in the next 2 days. Before I go into what we're actually going to be doing, Cindy, did I cover sort of the gist of the rationale?
Yeah, I mean, I think to emphasize the point, Florian, that you made, that the FDA doesn't really care in the end what the biochemistry looks like, nearly so much as whether it makes patients better and whether patients, we can demonstrate that patients are actually better. And the only way we can do that is to have some outcome measures the patients tell us that is better or worse. And I think that's kind of what we were going by when we did the surveys. For people who haven't completed or had a chance to complete surveys, maybe they're newly diagnosed, we actually, Jean Xeroxed and brought some of those, and we're happy to hand them out. If we run out of copies, we can either A, make more, B, we can send them to you electronically on a Word document by email, and you can fill them out. But I think it's the kind of information that is incredibly useful. We have not yet really tabulated all of the late onset Tay Sachs and Sandhoff surveys. Florian has, actually I should pull out here as an example of what it looks like. Florian and Annette Bly and the folks at Mass General really put together an excellent piece on the natural history of GM2, infantile GM2 gangliosidosis. In 2011, based on these surveys, we have yet to put something out like this for the late onset patients, but your input would be great. Lily's working on it.
Always helps to bring your coordinator to these sessions to add urgency to their work. So we are as Cindy said, I think we made quick success with the infantile surveys, and we really want to follow up on that now with the late onset. And I think it very nice dovetails with these kinds of prospective sessions that we're having in the next 2 days. So what kind of assessments? So maybe I should just go around first and ask regarding the rationale of why we're doing this. Does this make sense? Do you have questions about this? Do you understand why there is this need to harmonize and do these clinical prospective assessments? Essentially, it's about being able to measure change and understand whether if we started a drug tomorrow, it could make a difference in the particular testing that we're performing. If we don't know what the range is of that test in any given patient population, we cannot know whether the drug is working or not.
Well, it's a silly question, I think, but because late onset Tay Sachs patients have such a range of where they are, how in the world can you tell if it's working for someone who was just diagnosed compared to somebody who's lived with it for 40 years? That was my question.
That's a great question.
Thank you.
So as I said, there are no silly questions, only great questions. So that's a very good one, and one we struggle with all the time. with it, and it's a question whether if we had the perfect drug, if we had that perfect drug that works on either the enzyme or the gene level, would it work the same across all phases of the disease? And the answer is probably no, right? It would probably work better in certain stages, but not in others. And we know that the disease in late onset Tay Sachs syndrome affects different parts of the nervous system. We don't even know if the drug would work better for certain symptoms, but not for others, right? So you're absolutely right that there are different stages, but there are also different symptoms, and some of them might be more amenable to an intervention. So there clearly are challenges, but I think there are also great opportunities, because if we understand that there's just one symptom that is more amenable to change, it's sort of like getting your foot in the door. If we can get our foot in the door and we can push, then it might be possible to have other symptoms over time also be addressed.
I think, for these really rare diseases, in some cases, there are clinical trial designs in which the patient can be their own control. So if you pick a particular outcome measure, you can look at the way what it is like for 6 months on drugs, 6 months off drugs, 6 months on drugs. So there are ways to try and look at to deal with the variability of where people are. So at this point in the natural history, we're sort of shotgunning, looking across people at all the different stages of their disease to see what measures really would make sense for them.
I think the important point is that you don't want to throw out a drug just because it doesn't work maybe in the most severe form of the disease. It might work well in the early stages. It might tell you that maybe you have to go up higher with the dose or do something else for the more advanced cases. But I think what we really want to understand is that kind of variability and what happens at different stages, and that will help us.
Does this have anything to do with, are you basing this on the last trial, Zaveska, where, there was a trial, I think it's Zaveska, where it worked better for people who were just starting the symptoms than for people who were already in throes of it? So you're using that as like a springboard. So I guess it was worth what we went through.
It works better on certain systems than others too.
Yes.
The same with paramethamine, the Daraprim also. That was that was a lower dose that was better.
No, and that's, I think miglustat is probably the good example of a study in which the outcome measure of quantitative muscle testing turned out probably not to be the best measure that we could have used, and that maybe speech, because there was improvement in speech in Dr. Kolodny's trial, that maybe that was a better outcome measure, but we didn't think to use it, or the folks that did the trial didn't think to use it as an outcome measure. So really our question in doing these very broad evaluations is, what's going to be the best functional test we can use that will give us the results, the most sensitive test that's going to give us the results we need? And it's a little bit of a guessing game. I'm sure you can tell, Genzyme has been through this many times with many different diseases, and it's tough. You have to, it's a bit of a gamble, but you have to try and understand the disease well enough to know what's the thing you're going to pick that's really going to show a difference. So, in a sense, part of the assessments we want to do over the next couple of days are quite broad. And the idea is, can we figure out which ones of these might really be the best outcome measure? And that's hard. Do you want to add to that?
Also adding, I think Florian talked about the patient relevance. So is that specific outcome measure, can we change through the time of the trial, which is relatively should be short time. So we want to look at an outcome measure that could change within, say, a year period. And we also want to see how relative it is from a patient perspective, because the regulatory authorities may want to also, okay, you improved X, is this truly relevant? And how do you show that it's relevant? So that's why we're doing all of these assessments, but we're also asking you about what impacts you the most, and we're trying to link these together.
Does that make sense? As a neurologist, we study many things. Often we're studying signs, such as heightened reflexes, and maybe our drug might diminish heightened reflexes. But is that meaningful for a patient, right? So I think that's where it's so important that this is a two-way street, and that you're giving us feedback about what is relevant. Whereas I think the insight from the Zaveska trial that maybe speech, articulation, clarity of speech was impacted upon, that is something that I think patients would care about in their daily interactions. So this is an ongoing process, and we're trying to learn here together with you. One thing that's different, I think, that should also be emphasized is that we're, as opposed to prior trials, trying to create an all-inclusive platform and repository where we collect information on outcome measures that can be shared across many different studies and hopefully will become the basis for many future trials. So one of the things that we always want to have is natural history to compare to when we're starting a trial. And often past assessments were done one particular way, or certain trials use some outcome measures, and in another trial, other outcome measures were used. We now have the advantage of using a particular platform called Neurobank, which has been used for 2 decades now in the ALS world. And ALS has had tremendous success in building trial sites across the world. There are 100 different trial sites that use Neurobank, and they have a very experienced set of people doing data management and project management. And they're helping us align all these outcome measures. And when you are consenting to these studies, you're consenting that your information will be anonymized, your privacy will be maintained, but the de-identified information can actually be shared with other researchers so that we don't have to keep on duplicating efforts, so that we don't always have to start again, and so that we can build on these efforts. So I think so the nitty-gritty of the next 2 days is going to be that we're going to let a series of neurologists loose upon you, right? Is that one way of putting it? So I will say that when we started doing this, we did this in sort of rather informal ways over the years, and last year, thanks to Jerry and others, we started to become more organized, and we had 2 neurologists at the session of clinical assessments, and now we have 4. And I think it really speaks to the interest that is coming to this disease, and the fact that many more researchers and physicians are recognizing there is a real chance of us developing treatments for late onset Tay Sachs and SANTOF if we do these assessments well and rigorously. So if we really understand how you are affected by this disease, and we can quantify this in ways that are reproducible, and different neurologists do the same exam and find the same thing, then truly, if you find a drug that changes these measures, then that drug must be doing something. Okay, so it's about helping us arrive at more certainty. So there will be four groups, I think, A, B, C, D, and I understand that Diana has assigned people to different groups. Do you want to speak briefly to that?